Digestive system in human. The digestive system in humans is responsible for breaking down food into nutrients that can be absorbed by the body for energy, growth, and repair. It consists of several organs that work together to ensure the proper digestion and absorption of food. The process begins in the mouth, where salivary glands produce saliva that contains enzymes to start breaking down carbohydrates. The food is then chewed and swallowed, passing through the esophagus into the stomach, where it is further digested by stomach acid and enzymes. Next, the partly digested food moves to the small intestine, where it is mixed with digestive enzymes and bile from the liver and pancreas to break down carbohydrates, proteins, and fats. The nutrients are then absorbed through the walls of the small intestine and transported to the liver for processing. The large intestine then receives the remaining waste materials from the small intestine, where water and electrolytes are absorbed and waste products are formed into feces. Finally, the feces are eliminated from the body through the rectum and anus. Overall, the digestive system plays a vital role in the proper functioning of the human body by breaking down food to provide essential nutrients and energy for growth, repair, and sustenance. Definition of terms. A digestion. Digestion refers to the process of breaking down food into smaller, simpler molecules that can be absorbed and utilized by the body for energy, growth, and repair. The process involves mechanical and chemical breakdown of complex food molecules such as proteins, carbohydrates, and fats, into their constituent components by enzymes and acids. Digestion begins in the mouth and continues in the stomach and small intestine, where the nutrients are absorbed and transported to the liver for processing and distribution to the body's cells. Overall, digestion is a fundamental process that enables the body to extract essential nutrients from food to maintain health and vitality. Absorption. Absorption refers to the process by which nutrients, water, and other substances are taken up or moved from the digestive tract and into the bloodstream to be transported to different organs and tissues throughout the body. Absorption occurs primarily in the small intestine, where nutrients are broken down further and absorbed into the bloodstream by small finger-like projections called villi and microvilli. Different types of nutrients are absorbed through different mechanisms. Carbohydrates and proteins are absorbed into the bloodstream through active transport mechanisms, requiring energy to move against a concentration gradient. Fats, on the other hand, are absorbed through passive diffusion, where they enter the bloodstream through the lymphatic system before entering the general circulation. In addition to nutrients, other substances such as water, minerals, and vitamins are also absorbed through specific mechanisms in the small intestine. The efficiency of absorption depends on various factors such as the type of nutrient, the health of the digestive tract, and the presence of certain diseases or conditions that may interfere with the absorption process. Overall, absorption is a critical process that ensures that the body gains essential nutrients and fluids necessary for optimal health and homeostasis. Assimilation. Assimilation refers to the process by which absorbed nutrients are utilized by the body's cells and tissues for growth, repair, and energy production. After absorption, the nutrients are transported via the bloodstream to different parts of the body, where they are taken up by cells and metabolized for various functions. For instance, glucose, which is derived from the digestion of carbohydrates, is transported to cells' mitochondria to produce energy in the form of ATP through a process called cellular respiration. Amino acids are used to synthesize proteins, which are essential for building and repairing tissues in the body. Fatty acids are used to produce cell membranes and certain hormones that regulate bodily functions. The utilization of nutrients is regulated by various hormones and enzymes that control the rate of biochemical reactions, ensuring that the cellular processes proceed at optimal levels to maintain homeostasis. Overall, assimilation is a critical process that converts absorbed nutrients into usable forms for various metabolic activities that support the body's functions and survival. Ingestion. Ingestion refers to the process of taking food or drink into the mouth, also known as oral intake, to initiate the process of digestion. This initial part of the digestive process involves chewing, grinding, and breaking down food into smaller particles that can be swallowed and further processed by enzymes in the digestive tract. Ingestion involves a complex sequence of physiological events, where the mouth uses muscles and structures such as the tongue and teeth to masticate food and mix it with saliva that contains enzymes such as amylase that begins the breakdown of carbohydrates. Once food has been sufficiently broken down, it is swallowed and passed down the esophagus into the stomach, where it is further mixed with gastric juices that break down protein and fats. Ingestion is critical in providing the body with the necessary nutrients and energy to maintain bodily functions and support growth and repair. Ingestion is also a sensory experience as the taste, smell, and texture of food can trigger various reactions that impact appetite, satiety, and overall health. Enzymes. Enzymes are biological molecules that catalyze, speed up, chemical reactions in living organisms. They are typically proteins, and each enzyme is specific to a particular reaction, substrate, or class of substrates. Enzymes lower the activation energy required for a chemical reaction to occur, thus making it easier and faster for the reaction to take place. This process occurs through the formation of enzyme-substrate complexes, where the enzyme binds to the substrate, and a chemical reaction occurs, resulting in the formation of products. Enzymes are essential components of metabolism and are involved in numerous processes such as digestion, energy production, and biosynthesis. Some enzymes are produced by the body, while others can be obtained from food sources or synthesized through genetic engineering techniques. Enzymes are regulated in different ways, including allosteric regulation, covalent modification, and feedback inhibition, to ensure that metabolic processes occur efficiently and under optimal conditions. Without enzymes, many of the chemical reactions required for life would not occur at a sufficient rate, leading to various metabolic disorders and diseases. Types of enzymes. Intracellular enzymes. Intracellular enzymes are one type of enzyme. Intracellular enzymes are enzymes that are synthesized and function within the cells of an organism. These enzymes play essential roles in the metabolic processes of cells, including cellular respiration, DNA replication, and protein synthesis, among others. Intracellular enzymes are produced by specialized organelles such as mitochondria, ribosomes, and the endoplasmic reticulum. Different types of intracellular enzymes include isomerases, hydrolases, oxidoreductases, transferases, and ligases, among others. Isomerases catalyze the rearrangement of atoms within a molecule to produce an isomer, while hydrolases catalyze the hydrolysis breaking down of chemical bonds through the addition of water molecules. Oxidoreductases transfer electrons from one molecule to another, transferases transfer functional groups of atoms from one molecule to another, and ligases join two molecules together. Intracellular enzymes are tightly regulated by the cells in which they are produced, and their activity is influenced by various factors such as pH, temperature, cofactors, and substrates, among others. Extracellular enzymes. Extracellular enzymes are another type of enzyme. Extracellular enzymes are enzymes that are secreted by cells and act outside of the cell, mainly in the extracellular matrix, to catalyze various biochemical reactions. Extracellular enzymes are produced by many different types of organisms, including bacteria, fungi, plants, and animals. In bacteria and fungi, extracellular enzymes play essential roles in the breakdown of complex molecules into smaller, more accessible compounds that can be absorbed by the cell for energy and biosynthesis. In animals, extracellular enzymes have various functions such as digestion, blood clotting, and the breakdown of cellular debris. Different types of extracellular enzymes include proteases, lipases, amylases, cellulases, pectinases, and chitinases, among others. Proteases break down proteins into smaller peptides and amino acids, while lipases break down lipids, fats, into fatty acids and glycerol. Amylases break down starch into simple sugars, cellulases break down cellulose into glucose, pectinases break down pectin, and chitinases break down chitin. Extracellular enzymes are often synthesized and secreted under specific conditions and are regulated by various factors such as pH, temperature, substrate availability, and enzyme inhibitors. Naming of enzymes. 1. Trivial naming. Trivial naming is one of the methods used to name enzymes. This method of enzyme naming is based on the source or function of the enzyme or on its historic name. The names of enzymes based on trivial naming often do not provide much information about their catalytic properties or the nature of the reaction they catalyze. Instead, they may provide a general indication of the substrate the enzyme acts upon or the activities of the enzyme, such as protease, lipase, or kinase, among others. Examples of enzymes named using trivial naming include lactase, which catalyzes the breakdown of lactose, and hexokinase, which phosphorylates glucose during glycolysis. Despite their limitations, trivial names are often used in scientific literature and are useful in communicating information about enzymes to a broad audience. However, to avoid ambiguity, enzymes will often also have systematic names reflecting their systematic classification and reaction chemistry. 2. Use of suffixes. The use of suffixes is another method used to name enzymes. Enzyme names based on suffixes provide information about the enzyme's catalytic properties, reaction mechanism or classification. The suffixes used in enzyme naming often indicate the type of reaction that the enzyme catalyzes, such as ASE for enzymes that catalyze a specific biochemical reaction, including oxidase, kinase, and hydrolase. Other suffixes reflect the substrate specificity, such as lase for enzymes that catalyze the formation or rupture of a particular bond, including phospholipase, amylase, and pectinase. The suffix synthase is used for enzymes that synthesize new compounds, such as ATP synthase, which synthesizes ATP molecules, and glucosamine synthase, which synthesizes glucosamine. In addition to suffixes used to name enzymes, prefixes such as di, re, iso, and cis may be used to denote changes in the enzyme's reaction mechanism or specificity. The use of suffixes in enzyme naming provides a systematic way to name enzymes, allowing scientists to classify enzymes based on their properties and functions. Furthermore, using standardized enzyme names helps avoid confusion between different types of enzymes and promotes efficient communication in scientific research. Factors that affect the rate of enzymatic activities. Several factors can affect the rate of enzymatic activities. Some of the most important factors include 1. Substrate concentration. Increasing the substrate concentration can increase the rate of the enzymatic reaction, up to a point. At high substrate concentrations, the rate of the reaction may plateau due to the saturation of enzyme active sites. 2. Enzyme concentration. Increasing the enzyme concentration generally increases the rate of reaction, as more active sites are available for substrate binding. However, again the rate may plateau due to saturation of substrate. 3. Temperature. Enzymes have an optimal temperature at which they work best. At this temperature, there is maximal enzyme activity, but if temperatures go beyond this range, either by increase or decrease, the enzyme may become denatured or inactive, reducing the rate of reaction. 4. pH. Like temperature, enzymes have an optimal pH range in which they function at their maximal activity. pH outside this range also lead to denaturation or reduced enzyme activity. 5. Presence of inhibitors. Presence of inhibitors, such as competitive or non-competitive inhibitors, can reduce the rate of an enzymatic reaction by blocking the active site of an enzyme, or making the enzyme less active. Types of enzyme inhibitors. Enzyme inhibitors are molecules that bind to enzymes and reduce or abolish their activity. There are several types of enzyme inhibitors, including 1. Competitive inhibitors, compete with the substrate to bind at the active site of the enzyme. The structure of competitive inhibitors is similar to the substrate, so it can bind and occupy the active site, preventing substrates from binding, thus reducing enzyme activity. 2. Non-competitive inhibitors, bind to a site on the enzyme other than the active site, also called an allosteric site. Binding here changes the enzyme's shape and reduces the catalytic efficiency of the enzyme. 3. Uncompetitive inhibitors, also called allosteric inhibitors, bind to the enzyme substrate complex and prevent enzymatic activity. These inhibitors require both the enzyme and substrate to be present for inhibition to occur. 4. Mixed inhibitors, bind to both the free enzyme and the enzyme substrate complex, but with different affinities, resulting in an uneven or mixed pattern of inhibition. 5. Irreversible inhibitors, a type of enzyme inhibitor that cannot be removed and result in permanent or prolonged inhibition. They covalently modify the functional groups of the enzyme, rendering it inactive. Understanding the different types of enzyme inhibitors is important for drug design and optimization of enzymatic reactions in biotechnology, medicine and other scientific fields. 6. Presence of cofactors. Cofactors such as metal ions or coenzymes can activate or enhance the activity of an enzyme. 7. Enzyme specificity. Each enzyme is specific to a particular substrate or group of substrates, thus enzymes will only catalyze reactions with specific substrates that fit their active sites. Understanding the factors that affect the rate of an enzymatic reaction is important to optimize enzymatic processes for various applications, including biotechnology and medicine. Properties of enzymes. Enzymes are biological catalysts that speed up the rate of biochemical reactions in living organisms. They have several properties that make them unique, including 1. Specificity. Enzymes are highly specific, each one catalyzing a particular reaction, often involving a specific substrate. They have active sites with unique shapes that fit the substrate like a lock and key. 2. High catalytic efficiency. Enzymes typically increase the rate of chemical reactions by a factor of 10^8 to 10^14, making biochemical reactions occur rapidly. 3. Regulated activity. Enzyme activity can be regulated, either by feedback inhibition or environmental factors such as temperature, pH, or presence of inhibitors, enabling organisms to respond to changes in conditions. 4. Sensitive to environmental conditions. Enzymes are highly sensitive to environmental conditions such as temperature, pH, and salt concentration. These factors can affect their activity via altering their shape or structure. 5. Reusable. Enzymes are not consumed during a reaction and can be reused, which makes them highly efficient catalysts. 6. Generally protein-based. Most enzymes are proteins, but some enzymes are nucleic acid-based and called ribozymes. These properties make enzymes essential molecules in biological systems for the metabolism, growth, and reproduction of all living organisms. These properties also make enzymes invaluable tools in industries such as biotechnology, pharmaceuticals, and food processing, where their specific and efficient catalytic properties are used to drive chemical reactions at industrial scales. Importance of enzymes. Enzymes are essential molecules in biological systems and have a wide range of functions that are vital for life. Some of the importance of enzymes are: 1. Catalysis of metabolic reactions. Enzymes catalyze metabolic reactions, making the conversion of complex molecules into simple substances more efficient and easier, such as digestive enzymes that break down food into digestible compounds. 2. Control of biochemical reactions. Enzymes control the rate of biochemical reactions and are able to switch them on or off as required, such as enzymes that regulate the Krebs cycle in the mitochondria. 3. Specificity and selectivity. Enzymes are highly specific and selective, catalyzing only specific reactions, which minimizes the formation of unwanted side products. 4. Enzymes are widely used in industry. Enzymes are used in a wide range of industries, including food processing, pharmaceuticals production, textiles, and biofuels industry, to catalyze desired reactions without the use of harsh chemicals. 5. Medical and clinical applications. Enzymes are used in medicine, such as to diagnose certain diseases, or to break down specific proteins in diseases such as cancer. 6. Genetic regulation. Enzymes also play a role in genetic regulation, where they modify, cleave or polymerize macromolecules, such as DNA or RNA, which is crucial in regulating gene expression in cells. Overall, the importance of enzymes cannot be overstated as they are essential for biological processes, industrial applications, and medical treatments. Practical activity. Aim to investigate the effect of pH on enzyme-controlled reactions. Materials. Enzyme solution e.g., amylase. Starch solution. Buffer solutions of different pHs e.g., pH 3, 5, 7, 9, and 11. Test tubes. Timer. Iodine solution. Pipettes. Procedure. 1. Label 5 test tubes with the pH of each buffer solution. 2. Add 2 milliliters of each buffer solution to its corresponding test tube. 3. Add 2 milliliters of the starch solution to each test tube. 4. Add 2 milliliters of the enzyme solution to each test tube. 5. Start the timer and allow the reactions to proceed for a set amount of time, e.g., 1 minute. 6. Use a pipette to remove a small amount of the reaction mixture from each test tube. 7. Add 1-2 drops of iodine solution to each small sample taken in step 6. Observe the color change. 8. Record the time and color change observations for each pH level. 9. Repeat the experiment 3 times to generate average data. Results. The results should indicate the effect of different pH levels on the rate of enzyme-catalyzed reactions. For example, as pH increases, the enzyme activity may increase up to the optimum pH, the pH at which the enzyme is most active, and then decrease as the pH becomes too basic or acidic, leading to enzyme denaturation or reduced activity. Conclusion. Based on the results, it can be concluded that pH plays a significant role in enzyme function, and that enzymes tend to be most active within a particular pH range. The optimal pH range for the enzyme being tested can be determined from the experiment, and can be useful in further investigations or applications, such as optimizing enzyme conditions in industry or tailoring medical treatments based on enzyme activity. Practical activity 2. Aim. To investigate the effect of temperature on enzyme-controlled reactions. Here are the remaining steps for the practical activity. Materials. Amylase solution. Starch solution. Iodine solution. Water bath or hot plate. Test tubes. Thermometer. Dropper or pipette. Method. 1. Label 4 test tubes A, B, C, and D. 2. Into each tube, add 2 milliliters of starch solution. 3. Into each tube, add 2 milliliters of amylase solution. 4. Place tube A in a water bath at 0 degrees Celsius. 5. Place tube B in a water bath at 20 degrees Celsius. 6. Place tube C in a water bath at 40 degrees Celsius. 7. Place tube D in a water bath at 60 degrees Celsius. 8. Allow the tubes to reach these temperatures for 5 minutes. 9. At the same time, add 4 drops of iodine solution to each of 4 wells in a spouting tile. 10. After 5 minutes, add 2 milliliters of the contents of each tube to the corresponding well in the spouting tile. 11. Using a dropper or pipette, mix the contents of each well thoroughly. 12. Observe and record the color change in each well. A blue-black color indicates the presence of starch, while a yellow-brown color indicates the absence of starch. 13. Repeat steps 9-12 2 more times. 14. Calculate the time taken for the disappearance of starch in each tube and record the results. 15. Plot a graph of the time taken for the disappearance of starch against temperature. Note: be careful when handling hot water and never touch the hot plate with wet hands, and always use a thermometer or heat-proof gloves when removing tubes from the water bath. Practical Activity 3. Aim. To investigate the presence of enzymes in living tissue. Here are the steps for the practical activity. Materials: fresh liver or other animal tissue. Hydrogen peroxide solution, 3%. Test tube. Forceps. Glass rod. Method. 1. Cut a small piece of fresh liver or other animal tissue using clean scissors and forceps. 2. Place the piece of tissue into a clean test tube and crush it with a clean glass rod. 3. Add 2 milliliters of hydrogen peroxide solution to the test tube. 4. Observe and record the results. 5. If enzyme is present in the tissue, bubbles of oxygen gas will be produced. 6. Repeat the experiment with different tissues, such as potato, apple, or celery. 7. Compare the results obtained with different tissues and record the observations. 8. Discard the used tissue and hydrogen peroxide solution safely. Note: be cautious when handling hydrogen peroxide, as it can cause skin burns or eye damage. Always wear safety goggles and gloves, and handle the chemical in a well-ventilated area. Do not ingest or inhale the chemical, and dispose of its safety in accordance with local regulations. Practical Activity 4. Aim. To investigate the effect of substrate concentration on an enzyme-controlled reaction. Here are the steps for the practical activity. Materials: amylase solution, starch solution, water, iodine solution, test tubes, droppers or pipettes. Method. 1. Label 5 test tubes A, B, C, D, and E. 2. Into each tube, add 2 milliliters of amylase solution. 3. Into each tube, add 2 milliliters of starch solution, except for tube A. 4. Add 2 milliliters of water to tube A instead of starch solution. 5. Mix the contents of each tube thoroughly. 6. At the same time, add 4 drops of iodine solution to each of 4 wells in a spouting tile. 7. After 2 minutes, add 2 milliliters of the contents of tube A to the first well of the spouting tile. 8. Using a clean dropper or pipette, transfer 2 milliliters of the contents of tube B to the second well of the spouting tile. Mix the contents thoroughly. 9. Repeat step 8 for tubes C, D, and E, transferring the contents to the corresponding wells of the spouting tile. 10. Observe and record the color change in each well. A blue-black color indicates the presence of starch, while a yellow-brown color indicates the absence of starch. 11. Measure the time taken for the disappearance of starch in each well, and record the results. 12. Repeat steps 6-11 for each tube 2 more times. 13. Calculate the rate of reaction for each tube by dividing 1 by the time taken for starch disappearance, and record the results. 14. Plot a graph of enzyme activity against substrate concentration. Note: be careful when handling chemicals and always wash your hands thoroughly afterward. Always use clean equipment and dispose of chemicals safely in accordance with local regulations. Practical Activity 5. Aim. To investigate the effect of enzyme concentration on an enzyme-controlled reaction. Here are the steps for the practical activity. Materials: catalase solution, hydrogen peroxide solution, 3%. Water. Test tubes. Droppers or pipettes. Method. 1. Label 5 test tubes A, B, C, D, and E. 2. Into each tube, add 2 milliliters of hydrogen peroxide solution. 3. Into tube A, add 2 milliliters of water instead of catalase solution. 4. Into tubes B, C, D, and E, add 0.1 milliliters, 0.2 milliliters, 0.3 milliliters, and 0.4 milliliters of catalase solution, respectively. 5. Mix the contents of each tube thoroughly. 6. Immediately after mixing, observe and record the volume of oxygen gas produced in each tube after 2 minutes. 7. Repeat steps 2-6 2 more times. 8. Calculate the rate of reaction for each tube by dividing the total volume of oxygen gas produced by the time taken for each reaction, and record the results. 9. Plot a graph of enzyme activity against enzyme concentration. Note: be careful when handling chemicals and always wash your hands thoroughly afterward. Always use clean equipment and dispose of chemicals safely in accordance with local regulations. Also, note that hydrogen peroxide can be dangerous and can cause skin and eye irritation, so wear gloves and goggles when handling it.